All right. Hey, folks. Thanks for joining us this Friday morning. My name's Joe Wood. I'm a senior scientist at the Chesapeake Bay Foundation. Super big fan of freshwater mussels. And this morning, we're excited to share a new tool that we've been working on for quite a while. Um, I've been interested in freshwater mussels, I think almost 10 years now, and particularly interested in the connection between freshwater mussels and Chesapeake Bay restoration work. And several years ago, we did a stack workshop. We had this conversation that centered around the question of, why are freshwater mussels not more connected to bay restoration? And I emerged from that, and I think many people emerged from that with th three kind of top 10, three takeaways for me from that was that first, like mussels are really cool and they're exciting. They have a lot of features. And they can be engaging. There's a lot of really interesting things about freshwater mussels. Number two is that they're good for our streams, whether it be their filtering capacity, their ability to create habitat or other things. They're an important part of our stream ecosystems. And three, that they're really imperiled, and they face a lot of emerging threats. Um, their numbers are declining. We think they've lost, 90% of their historic populations. And so, one of the things that we asked following that meeting was, how can we connect bay restoration work with freshwater mussels? And a specific question that came up is, if we wanted to do work that blended those two things, what would it look like? And forested buffers came up as a clear path forward. If you look at state wildlife action plans and you look at freshwater mussel species that are imperiled, oftentimes one of the most important things is, we need more riparian forested corridors. And so, what evolved this project with Joe and Shiva, Joe Weber and Shiva Turabian from the Department of Natural Heritage at DCR was really, I think, a good answer to the question of, what does our local freshwater mussel population look like? Where might the richest habitats exist? And if we want to protect those habitats, what should we do about it? Where do we go? Where can we implement practices? Where might we need to conserve forested buffers? And so they'll go through the technical details this morning. They've done really great work here. I did want to mention two other things. Many of the people on the call are from outside of Virginia. This tool is just the Bay watershed portion of Virginia. But we think that there's application for this type of approach Chesapeake Bay wide, and that it could be a helpful context to think about how do we manage for, the Chesapeake Bay restoration, but also at the same time think about our local waters and think about local living resources. So I'd be welcome any conversations with folks after this about how that might happen moving forward. The second thing, as a part of this grant, this work was funded by Chesapeake Wild, which just happened to open this week. So this is sort of a timely presentation. But another piece of this work was the development of 3D mussel replicas. And so if you look at my backdrop here, these are all 3D printed freshwater mussel replicas, and their digital files are open source. And so if anybody is interested in getting a set of those printed for education, for outreach, for training, I'm happy to help you do that offline at some point later. So with that, I'll hand it over to Joe and Shiva, and we'll take it away.
Thank you very much, Joe. You hear me okay? Great. I want to start by thanking Joe and Liz and the rest of the Chesapeake Bay Foundation for involving us in this awesome project. And I want to tell you that Shiva and I are very excited about being here today to present the products to you. Oh, of course, it doesn't advance now. It did, but now it doesn't. I'm going to stop sharing and try again.
So while Joe's getting that up, I will mention, so these tools that we're going to be presenting today are online through the NHDE page at the Department of Natural Heritage for people to use. And we'll circulate that link later today.
Okay, Did that advance for you, Joe?
It did, yeah.
All right. So Joe touched on this a little bit. Why do we care about mussels? Well, in the Virginia Natural Heritage Program, our business is biodiversity conservation. So this aligns very well with our mission. North America has the highest diversity of freshwater mussels in the world. There are about 300 species. And mussels are important components of diverse aquatic communities. We're also interested because freshwater mussels are the most imperiled group of animals in North America, with 66% of species being at risk. And mussels are declining due to habitat alteration, pollution, exotic species, and other factors. We're also interested in mussels because of integrity. They provide important ecosystem services and functions. They're filter feeders that help purify aquatic systems and make resources available to other species. They have been shown to enhance denitrification of fresh waters, reducing nutrient loads downstream, which is obviously very important for water quality in the Chesapeake Bay. They're an important food source for many species of wildlife. They not only maintain habitats, they are habitats for other species. You could think of a crayfish using a mussel bed for shelter. In general, healthy mussel populations equal healthy aquatic systems. There's been a lot of interest in mussel restoration because extirpations and widespread population declines reduce the benefits that we're talking about here. Why this project? Well, Joe talked about that briefly too. The Scientific and Technical Advisory Committee of the Chesapeake Bay Program, which is supposed to provide guidance on measures to restore and protect the bay, met in a workshop in 2020 and chose freshwater mussels as the focus of that workshop to consider ecosystem services, to document biodiversity, to outline the intersections with other Chesapeake Bay issues and identify opportunities for multiple benefits, and to explore their potential to engage partners. That report recommended protection and restoration efforts, community engagement, support for partner agency surveys, and the additional of mussel surveys to monitoring programs. It asked for more science, more study. And they wanted an evaluation of the conflict between nutrient management and mussel restoration. And soon after that report was released, Joe, who was the lead author, contacted us to request that we map forested buffers that prioritize freshwater mussel outcomes. So how did we model buffers for freshwater mussel habitat? We started by developing a species habitat model for each of the 19 native freshwater mussel species that occur in the Virginia Chesapeake Bay watershed. This was unusual for us because we typically only work with rare species, but for this project, we modeled common species as well, and I'll talk about that more. Then all of those 19 models were stacked to create a comprehensive approximation of native mussel biodiversity. That's the mussel richness map. We integrated reaches of high richness and rarity from the mussel richness map with information on overland flow, nutrient loading from non-point sources, and land cover characteristics to strategically delineate and prioritize buffers for conservation and restoration of mussel habitat. I'll start with the methods we use for species habitat modeling. It all begins with training data. We collected, prepared, and reviewed data from multiple sources, including our element occurrences. These are polygons for rare mussels tracked by the Virginia Natural Heritage Program, which we maintain in our biotics database. We also included watch list species. These are species for which we did not create element occurrences because we don't know that they are rare. We're just keeping an eye on them. If they do become rare enough for us to track them, then they're going to become element occurrences and be included in the regular database. But they were included in this project. We also conduct mussel surveys in our program, so we have a database resulting from those mussel survey reports. We use the Department of Wildlife Resources Species Observation Database and also their extremely comprehensive survey data. We ran scripts to QC, align, merge, and clean data sets. We associated observations of presence and absence with reaches from the National Hydrography data set. And this is another benefit of using those mussel survey reports. They provided data that we typically don't have. They allowed us to train models for common species. We usually don't have point data for common species. And really interestingly, they allowed us to use true absences in our modeling. Usually we have to create what's called pseudo absences, where we randomly and sometimes stratify points throughout our study area, making sure that none of those points overlap with known locations so that we don't confound the models. And then we use those as pseudo absences. It works pretty well for rare species because they don't occur all over the landscape typically, but common species do. So this allowed us to use true absences in our modeling work, and that leads to better predictions and also better validation metrics. So now I'll talk about the environmental predictive variables, and the preparation of these environmental variables is typically the largest part of a modeling project. You can spend 60 to 80% of your time developing your environmental predictive variables. We attributed the training data set, those presence and absence reaches, with environmental variables from 4 sources. The primary source was StreamCat, which included variables for climate, geology, topography, land cover, and agriculture. And there also was a variable for the presence of dams. We supplemented this with an additional set of predictors summarized at the catchment level for select climate, topography, land cover, and soil variables. We use the NHD plus medium resolution data set for stream flow and velocity. And then we use this non-typical data set for environmental modeling. The Oak Ridge National Laboratory hydrosource data set was actually developed for hydro energy, but it was very useful for variables on stream temperature, substrate, and hydrological attributes. Variables that were not correlated with at least one species' presence or absence were removed from the modeling for that species, which makes sense. If it's not predictive, there's no reason to have it in there and to cause further confusion in the modeling. And then for correlated variables, we removed all but one variable from correlated groupings of variables. Okay, now we're ready to get into the modeling. For this project, we use a random forest classification approach to associate known presence and absence reaches for each mussel species with a set of environmental predictors. Random forest is an ensemble classifier in that it uses a large number of individual classification trees, each built from random subsets of training data and environmental predictors. And then it aggregates the results across all the trees to develop a final model. And this is where the real power in this methodology comes out, because we're running the model over thousands of iterations to create thousands of classification trees. And then each one of those classification trees is developed from a different randomized set of training data and environmental variables, which it seems kind of crazy because it doesn't make any sense to do that. But that randomization combined with the number of iterations ends up giving you very robust predictions. And it identifies associations that the human mind just wouldn't have come up with. And that sometimes experts doubt, but end up to be real in the real world. So we use a jackknife cross-validation technique for validation. This is also called leave one out, in which a single group of presence and an equal number of absence points are held out for each iteration, and then you see how well the model performs. This yields a set of validation metrics and model thresholds, which you see in the legend on the lower right. And then your results are predictions of habitat suitability over all reaches. So this is what's called the voting process, in which the proportion of trees which are classified as present are the votes for whether or not the habitat is present there. So you take your thousands of models and you stack them on top of each other, and you look through a single cell And then each time that cell is voted present in the individual trees, that is your probability of suitable habitat at that location in your final model. And then we generate a comprehensive metadata sheet for each model. After the models were developed, we distributed them for internal and external reviews on ArcGIS Online, where we provided instructions and asked the reviewers questions. We asked them to select thresholds for predicted suitable habitat and also to suggest any modifications as necessary. We then applied those thresholds to the models and modified them to, as recommended, to produce predicted suitable habitat layer for each species. And there's an example of one there. And then here's the mussel richness map, which we created by overlaying the predicted suitable habitat summaries, the predicted suitable habitat layers for each species to summarize them and produce a map of total mussel diversity. The legend indicates the number of species represented by each class. The classes with rarities means that the habitat of at least one mussel tracked by the Virginia Natural Heritage Program was predicted there. We then developed buffers for conservation and restoration of significant mussel habitats. What did we determine was significant? Well, any reach that was found in the top class of 7 to 13 predicted habitats in the darkest blue, or if they contained any habitat of rare mussels, so any of the shades of magenta. I'll now turn the presentation over to Shiva, and she'll explain how the buffers were developed.
Thank you, Joe. Hi, everyone. How we develop conservation and restoration buffer. Follow buffer were developed using 4 inputs, including significance, reaches for the mussel richness map, Virginia land cover developed by Virginia Geographic Information Network, high resolution national hydrography plus dataset, and 2022 non-point source pollution assessment developed by DCR. Next, please. From mussel richness map, 4 classes with rarity and high richness were used to develop restoration and conservation buffer. As you can see, the classes with 1 to 3 species with rarity, 4 to 6 species with rarity, 7 to 30, that is high richness class of mussel richness map. The Virginia land cover layer includes 12 classes, which we use just natural and agriculture category for our analysis. Next, please. Accordingly, the natural cover include forest, harvested area, shrubland, tree cover, and wetland, while the agriculture cover consists of cropland and pasture, that we use just these classes for our analysis. The non-point pollution dataset from the, DCR includes estimated loss of nitrogen, phosphorus as sediment for each hydrological units that we converted the estimated nutrient level into a score ranging from 0 to 100. After that, we combine these nutrient level layer to create an overall level map, nutrient level map, where areas were classified as low if all nutrient level were low, medium if at least one nutrient level was medium, and high if any or just at least one nutrient level was high. As you can see, again, in each hydrological level, we have the map with high, medium, and low nutrient level. To create inner and outer buffer around the area of extracted mussel richness, we consider slope, soil sensitivity, and headwater condition. Inner buffer range from 100 to 200 feet, while outer buffer extended from 200 to 400 feet. to create the buffer around the flow line and streams. We classify the created buffer based on land cover. That buffer located in natural area were designated for conservation, while those in agriculture area were classified for restoration. To prioritize the conservation and restoration classes, we used overall nutrient level. That area with low and medium nutrient level were classified as medium priority of conservation and restoration, while if these areas were located in high nutrient level, were classified as high priority of restoration and conservation. And all buffers are important, so we opt to not to classify any low priority. At the end, we identify 4 priority classes for conservation and restoration. As you can see in the map, we have high priority of conservation and restoration, and medium priority of conservation and restoration. Accordingly, the largest area fall under medium priority of conservation with over 100,000 acres, and a smallest area is classified as high priority of restoration. And now I will just turn the presentation back to Joe to discuss use and access to these tools.
Thank you, Shiva. So I want to summarize how these products can be used. The mussel richness map will be used to inform additional field surveys to increase our understanding of mussel distributions and provide more data for training models in the future. It'll inform stocking efforts. So we're talking about seeding mussels raised in hatcheries into these reaches of waters. They're going to inform conservation planning, such as for the statewide freshwater mussel conservation plan under development in the Department of Wildlife Resources. The flow buffers will be used to design vegetative buffers on the ground. So in most cases, we're talking about planting trees. They'll be used to target lands for agricultural best management practices. So in addition to buffers, there are other management prescriptions to reduce sediment and nutrient loads into waterways. So example, farmers can use cover crops, they can keep livestock out of waterways, etc. And finally, to target lands for conservation. This does not necessarily mean acquisition. Rather, landowners can enter agreements to maintain vegetative buffers, and if the lands are in agricultural use, farmers could agree to use best management practices. The Mussel Richness Map is available on the Virginia Natural Heritage Data Explorer. No subscription is necessary to view these data. Just enter the address that you see up here on the top, vanhde.org, and then find a layer in the table of contents under the Conservation Planning group. And then literally as of this morning, the buffers are also available on Natural Heritage Data Explorer. They can be found just above the Mussel Richness Map in the table of contents. And again, I want to thank the Chesapeake Bay Foundation for involving us in this awesome project, and also the National Fish and Wildlife Foundation for funding. Thank you.
Great. Thank you guys so much. We've got plenty of time for questions if folks have them. I don't see anything in the chat so far. I guess I'll just kind of have a closing statement along the lines of, we think this is a really powerful tool in the sense that if you're doing buffer work, if you're doing work throughout the watershed and thinking about restoration and how to prioritize, it's a clear map for where should we go if we want to protect important freshwater mussel communities. So we think that could be really valuable moving forward. The second piece is that we think this gives us a little bit more of a comprehensive understanding of where mussels exist in the Bay watershed. If you're thinking about propagation and release of freshwater mussels, this is probably maybe a first step to think about what you might think there. Certainly some other, guardrails and things that you need to go through. But again, we think it's going to help help us move forward on freshwater mussels in Virginia, and hopefully we can do something like this across the Bay watershed. So I'll pause there for questions. If you want to come off mute, I think we can make that happen, or you can just put it in the chat. Give it another. I'll give it another minute here. Don't be bashful. Scott Heidel. Liz, are you able to unmute him?
Hello, thank you very much. This was an outstanding presentation. I really appreciated hearing it. Can you all hear me?
Yeah, we can.
Okay, excellent. So we're doing a lot of restoration work in Pennsylvania as well, and we would love to couple that work with the introduction or reintroduction of extirpated species where possible. So anything that we can do to get linked into whatever hatchery work is happening and proposed repopulations, be highly interested if we could stay in communication on that.
Sure. Yeah, great. Thanks, Scott. Joe, there's a question in the chat from Aaron Riley about several sites in the York salty water. You want to talk about that a little bit?
Sure. So when we first conducted this modeling, we looked at that and I was like, yeah, there seems to be a few species that go all the way down to the Chesapeake Bay, and they're supposed to be freshwater mussels. Brian Watson at Department of Wildlife Resources said there are indeed freshwater mussels that go that far down to the bay. So those are legitimate predictions. I also want to say that they are predictions, right? So a famous statistician said, all models are wrong, but some are useful. So you can't expect to zoom in on every area and for it to be absolutely correct that there are 6 species of mussels in that area. We're predicting suitable habitat. So there are multiple reasons why a mussel may or may not occur in a reach of river anymore. The habitat could look perfect now, and the mussel might be gone because of a past pollution event that happened decades ago, and the mussel just hasn't been able, mussel and or its host haven't been able to get back into that water. So we're modeling current conditions, and we're identifying habitats that would support the species, whether or not they actually occur there presently.
I think I'll just follow that up with the fact that a huge driver for the outcome of this was the input of all the observations we have of freshwater mussels in Virginia. And so I think the outcome is really closely connected to where we have seen mussels in the past. And, I think that's important to keep in mind. We got a question from John. York, PA is definitely looking at restoring mussels. Preliminary surveys have found mussels in the most unexpected habitat, or worse, in Center City, York, USA, CE flood project. The mussel experts were very surprised that such habitat had the most mussels we found. Just info that mussels exist in the worst expected habitat. Yeah, absolutely. So starting with the data, I think is always helpful. We don't always know what things look like. Great. Well, not seeing any more questions, I'll just say thanks so much for joining us this morning. This was recorded and will be posted for anybody that's interested in using it. If you have follow-up questions about how to use it or the replicas or anything else, please feel free to reach out and happy to help walk you through that. But thanks again, and I hope this is useful to you.